Bismillah Rahman Rahim, June 24, paper 2-2, and this is the first video. The time is 1 hour 15 minutes, and you've got to be very careful in seeing that you finish the paper well in time. The first question is now about smooth muscles, and smooth muscle is a tissue composed of smooth muscle cells. The cells contain cytoplasm packed with proteins that are involved in contraction and relaxation. That's a very important thing. Muscles can only contract and relax. Please do not ever write the word stretch and recoil. That is not for muscles, that's for elastic tissue. Smooth muscle is present in the airways of the gas exchange system. So they're telling you this in the question. Explain how smooth muscle cells in the walls of the bronchioles contribute to the function of these airways. So basically, muscles can contract and relax. That gets you 1 mark. Now, when they contract and relax, now what you've got to understand, if it contracts, the diameter becomes smaller, the lumen diameter becomes smaller. And when it relaxes, it goes back to its original diameter. So this is the bronchiole, this is the wall of the bronchiole, right? So if it was wider, so then it was this wide. Now when it will contract, the lumen will become smaller. So diameter size can be controlled, or you can say changed. The lumen diameter size. The lumen diameter size, please remember, diameter or the lumen size can be controlled or changed. Then this will control what? This can also regulate the flow of air or the volume of air flowing in and out. So if it becomes narrower, so less volume of air flows in. So it will regulate the flow of air, or you can say the volume of air flowing. You can't say it regulates oxygen out and carbon dioxide or something like that. You're not going to talk about that. So it is just going to regulate the flow of air, or you can say, I don't think you can say anything else because everything else is not allowed. You can't say oxygen and carbon dioxide. It's air which is flowing in and out. So when these smooth muscles will contract, another fourth mark scheme point is when the smooth muscle contracts, it reduces the air movement, or you can say it prevents the entry of contaminants. Normally it is relaxed, but when it contracts, this causes constriction when needed. Sometimes it is needed. But suddenly, if there's cold air entering, the smooth muscles will contract, so less cold air because it can really be tough on the lungs. So 2 marks, and there are 4 mark scheme points, and any of those two, any two out of those four, you could have given me, and I would have given you 2 out of 2. So contracts and relax 1 mark. Diameter size can be controlled, or you can say changed. Diameter or the lumen size can be controlled and changed. Then regulates the flow of air and contracts to reduce air movement. You can just see how this has got to be worded. So just pause the video here and have a look at this. Now coming to part B of the question, when viewed in longitudinal section LS, smooth muscle cells are elongated and taper at both ends. This is known as the fusiform shape. Each cell has a central nucleus which also appears elongated. Figure 1.1 is a diagram of a smooth muscle cell to show the fusiform shape. Now they can say the width is 5 to 10 micrometer and the length is 30 to 300 micrometer. A student used a microscope fitted with a calibrated eyepiece graticule to estimate that the length of one smooth muscle cell was 250 micrometers. Name the type of microscope slide that the student used to calibrate the eyepiece graticule. Well, that's simple. It was a stage micrometer. So stage micrometer will help to calibrate the eyepiece graticule. Now coming to part 3, it says the smallest object the student can see without the use of microscope is 0.2 millimeter in length. Explain whether the student would be able to see a length of 250 micrometer without the use of a microscope. Well, you see 0.2 millimeter, 0.2 millimeter is equal to 200 micrometer. So 0.2 millimeter is the smallest that the student can see. This is less than 0.25 millimeter because 250 micrometer is 250 is equal to 0.25 250 micrometer is equal to 0.25 millimeter. So you'll have to be really, student can see less than 0.25 millimeter. The cell is 50 micrometers longer than 0.2. 0.2 is 200, but this cell is 250. So the cell is 250. So you've got to give me a reason why. What can be the reason? You see, they've said in the question that the student can see without the use of microscope something which is 0.2 millimeter. 0.2 millimeter is 200 micrometer, but this is 250, so it's bigger than that. So yes, of course they'll be able to see it. Yes, we'll be able to see. Like for instance, if you can see something which is 200 micrometer without a microscope, so something which is bigger than that, that's of course then you can see it. Now coming to question number part C, figure 1.2 is a photomicrograph of smooth muscle tissue in the wall of the intestine. A capillary is visible. A capillary is visible in addition to smooth muscle cells. Oh, it's a little vague. Now it says outline the features that help to identify the blood vessel in figure 1.2 as a capillary. This is the one which they are wanting us to talk about. This thing here which is going like this, and you can see the single file of red blood cells. So outline the feature that helps to identify the blood vessel in figure 1.2 as a capillary. Now part 1 of the question, it says outline the features that help to identify the blood vessel in figure 1.2 as a capillary. Basically, it's very simple. The wall of the capillary is 1 cell thick. The wall of the capillary. You can't say capillary is 1 cell thick. This is another mistake that you write. The wall of the capillary is 1 cell thick, not the capillary by itself. So the wall of the capillary is one layer or 1 cell thick, and the other factor would be the second mark scheme point would be red blood cells transported in a single file. So that means the capillary is very narrow. And the size of a red blood cell is 7 micrometers. So that means these are even just a little bigger than that, the volumen of it. So red blood cell diameter, same as capillary lumen. Or you say diameter of the capillary narrower than smooth muscle cell. And no muscle and elastic layer in it. Capillaries do not have any muscle and elastic layer in it. So you have to give me 2 marks, and I have given you about 4 mark scheme points, 5 in fact, and any 2 out of those. So many points that you could have given me and you could have got your 2 marks. Wall of capillary 1 cell thick, RBC transported in single file, RBC diameter same as capillary lumen, no muscle and elastic layer, diameter of capillary narrower than smooth muscle cell. Part 2, explain how the structure of a capillary is related to its function in smooth muscle cell. Basically, they want to know if you know the function of a capillary. So basically, what is the function of a capillary? It is going to allow the substances to pass out of the blood. And what are those substances? Oxygen, glucose, amino acids, cell signaling molecules. Cell signaling molecules will be, that is going to be supplied to the muscle cell, and all the waste products will be removed, like carbon dioxide, lactic acid, or any other waste. So basically, it's going to be exchange of gases like glucose in, CO2 out. Capillaries only allow that. Arteries don't allow that. Arterioles don't allow that. Venules don't allow it. Veins don't allow it because they have thick walls. The other, you can say thin wall, so short distance. You see, the cell is just outside it. And because the capillary wall is very thin, so it's a very short distance through which oxygen and glucose has to pass, or glucose or oxygen or amino acids and cell signaling molecules have to pass. Number 3, in the endothelium is the lining of the capillary that has pores, or the word is fenestrations. You can also use the word gaps. So endothelial, you can say pores. So you can see these words, but you please do not come up with other words which I cannot give you marks for when I'm checking the papers. So endothelial pores, or you can say fenestrations, that's a difficult English word. Maybe you know it, maybe you don't know it. For more efficient passage. Then the endothelial pores will also allow passage of phagocytes, or you can say macrophages or neutrophils. Then the fifth point is narrow diameter. So all the cells are very close to it. Narrow diameter. So to reach the cells, or you can say all the cells are very close. Then the fact that the red blood cell is carrying oxygen and a bit of carbon dioxide. So the narrow lumen slows the red blood cell's movement for the uptake of oxygen. Or you can say slows the movement to increase the time for exchange of gases. Only one red blood cell fits at a time, so there is short diffusion distance, as the red blood cell is very close to the cells for gas exchange. So, I mean, this is structure to function. Structure to function, but basic function of the capillary is what? To allow oxygen, glucose, the exchange of substances. The smooth muscle cell needs oxygen, needs glucose, needs amino acids. How is it going to be supplied by the blood is going to carry it, and in the capillaries it's going to diffuse out into the muscle cell. So that's happening everywhere in the body. It's not just the muscle cell. They just want to know the function of a capillary. So a lot of mark scheme points for you to answer this question. There are about 1, 2, 3, 4, 5, 6 points. So supply. Let me just change this. Supply oxygen, remove carbon dioxide. Thin walls, so short distance. Short distance, then they're not very away, they're not far away from the cells which they are providing, the smooth muscle cell. Thin walls, so short distance. Endothelial pores, so more efficient passage. Endothelial pores for passage of phagocytes. When phagocytes move out of capillaries, when there is a site of infection. Narrow diameter, so is able to reach the cells. Narrow lumen slows the RBC movement for the uptake of oxygen. You see, because of the narrow lumen, then the red blood cells have to go in a single file and they go to go in a line. Now, this slows down their movement, but that is good for them because they can then, the uptake of oxygen is very, very efficient. Now, I always say this is all very good English. If your English is good, you can do very well in the bio exam. But I'm afraid your English is poor, you're not going to do very well in the bio exam or any exam. Part D of the question. Caldesmon is a large protein with a number of binding sites to attach to other proteins. So it's a large protein with a number of binding sites to attach to other proteins. Caldesmon exists in two different forms, H-caldesmon and L-caldesmon. H-caldesmon helps to regulate contraction and relaxation in smooth muscle cells. L-caldesmon is found in some non-muscle cells, where it also acts as a regulatory protein. Caldesmon is coded for by a gene known as CALD1. CALD1 has 17 exons. The primary structure of H-caldesmon has a repeating sequence in the middle of the amino acid chain that is not present in caldesmon. Repeating means the same amino acid comes again and again and again and again, but this does not happen in the other one, in the L-caldesmon. Researchers have discovered that a gene mutation is not the cause of the two different forms of caldesmon. Explain what is meant by gene mutation. Basically, what is meant by gene mutation is a change in the sequence of base pairs in a DNA molecule. This is the base pairs were what? The base pair was AT, GC, AT, AT, AT. And something changes and this one is removed. So the base sequence changes. So a change in the sequence of base pairs, it was AGAA, now it is AGA. So it is basically a change in the sequence of base pairs, and this will code for an altered polypeptide, or it will change the amino acid sequence, which is the primary structure of this polypeptide. So because remember there are 2 marks, so you've got to give me an explanation. You have to say, okay, a change in the base sequence, and then you have to say, okay, so this will result in a changed polypeptide, or it will change the primary structure of the amino acid sequence and resulting in a changed polypeptide. So change in the sequence of base pairs in a DNA molecule and change in the primary structures on altered polypeptide. Then we come to part 2 of the question. Researchers now know that the two different forms of Caldesmol are the result of events occurring directly after transcription of DNA. Now please remember, from DNA what do we have? The first thing we have is pre-mRNA, and then we have mRNA. What happens from pre-mRNA to mRNA? What is the difference? Here introns are removed and then the exons are put together. Now the exons can be put together in different combinations. Like there are 6 exons, so you have 1, 2, 3, 4, 5, 6, but you can have it 1, 3, 2, 4, 5, 6. So the exons can be then joined together in different combinations, and this is called gene splicing. So this is what you've got to understand is changes occur to the primary transcript that is formed by DNA transcription, suggest how the smooth muscle cells and non-muscle cells can process different forms of Caldesmol from the same primary transcript. So DNA is the same. This DNA is the same. But then what happens in pre-mRNA? Some changes occur. So the first thing is differences owing to RNA or gene splicing, removal of introns. Then second is exons joined differently in a different combination, and messenger RNA formed or the mRNA formed is different. So in one situation the mRNA is like this, in the other situation the mRNA is different. So the sequence of the bases is different in the mRNA. Here it is A U U A U. Here it is A U C C C G G G. So the sequence on the mRNA is different. So this will be a different mRNA and this will be a different mRNA. So differences owing to RNA splicing during removal of introns. There can be a difference. The introns, you can't say, please do not say it's a reject. If you say if different number of introns removed or some introns remain, that's wrong. Exons joined differently in different combinations. mRNA formed is different. Part 3 suggests how the two different forms of Caldesmol can still have similar functions even though they have a different primary structure. Now, of course, that is, how would they have the same, what will they have the same? They will have the same tertiary structure, or you can say the binding sites shape will be the same, or the removed amino acids are not actually structured amino acids, and removed amino acids are not part of the binding site. R group interaction is still the same. Now I explain this to you in a different manner. I say, okay, the active site is this. The active site is this depression on the surface of an enzyme. Now if this part changes and this becomes like this, well, it doesn't want to matter because the active site is the same. The active site should remain the same. The rest of the shape of the enzyme here can change, become something else, but it's not going to affect the function of this. So you've got to understand that. So similar tertiary structure or the binding site or the 3D shape is same. Then removed amino acids are not part of this area. They do not contribute to the active site. So removed amino acids are not structural amino acids. Removed amino acids are not part of the binding site. And the R group interactions are the same. You know, the disulfide bonds formed wherever there is, and they are the same. So this is what we had to give you for this 1 mark. So similar tertiary structure, removed amino acids are not part of binding site, and R group interactions are still the same. Question number 2, alveolar macrophages are cells of the immune system that remain in the alveolar region of the gas exchange system. The macrophages protect against infection caused by pathogens that have been inhaled. Alveolar macrophages have the same cell structure as typical animal cells. Complete table 2.1 to name the cell structure that match the function stated. Do not use abbreviations. Manufacture ribosomal subunits from proteins and ribosomal RNA, nucleolus. You either know this or you don't know this. Then synthesize triglycerides and other lipids, smooth endoplasmic reticulum. So you cannot say SCR. You cannot say SCR because it says do not use abbreviations. So smooth endoplasmic reticulum. Then pair of what? Function is organized microtubules of the cell cytoskeleton. So pair of centrioles. Now we come on to the part 2 of this. Figure 2.1 is a diagram of an alveolar macrophage showing some of the cell structure that be visible using an electron microscope. A newly formed phagocytic vacuole containing 2 cells of Mycobacterium tuberculosis. So they are showing you some of the cell structures, and they are showing you a newly formed phagocytic vacuole, phagosome, which is written here, containing 2 cells of Mycobacterium tuberculosis. The cell structures with the functions described in table 2.1 are not shown in figure 2.1. So we're talking of the ones which are in table 2.1 are not shown in figure 2.1. Complete figure 2.1 drawing and labeling the cell structure described in 2.1. Now you can see this is only for 2 marks. So if you drew all 3 of them were correct, you got 2 marks. If 1 or 2 was correct, you'd get 1 mark. All 3 structures should have been correctly drawn, and they must have been labeled. If you did not label them, then it's only 1 mark. But you drew them correctly. Coming to the part B of the question, TB tuberculosis can be prevented if the bacterial cells that have reached the alveoli are rapidly destroyed. Alveolar macrophages can detect the presence of M. tuberculosis in the alveolar space and carry out phagocytosis to form phagocytic vacuoles, such as the one shown in figure 2.1. Outline the sequence of events that leads to the formation of a phagocytic vacuole after detection of the bacterial cells by an alveolar macrophage. So basically, endocytosis occurs 1 mark. Bacterial cells, you can say chemotaxis, which is the chemotactic response when the bacterial cells antigens bind to the macrophage cell surface membrane. And then the third point was membrane fusion, vacuole pinches off. So sequence of events that lead to the formation of a phagocytic vacuole. Endocytosis, chemotaxis, vacuole pinches off. Part 2, name the cell structure that fuse with the phagocytic vacuole and release hydrolytic enzymes to destroy the bacterial cells. Lysosome. So that was simple. And then we come to the C part of the questions. Researchers have shown that vaccination programs are cost effective and very helpful in the prevention and control of TB. The program may be aimed at a particular group of people that are at a high risk of getting the disease, or they may be aimed at an entire population because the country has a high number of cases of TB. The Bacillus calmette-guerin BCG vaccine is freeze-dried and contains live, weakened, attenuated Mycobacterium bovis. Apart from being cost-effective, suggest and explain the advantages of using the BCG vaccination for the prevention and control of TB. We in Pakistan all have got the BCG vaccine. We must have got it when you were a kid. You must have got the BCG vaccination because in Pakistan TB is very common, so everybody gets it. Now the reason that it's what is the advantage of using the BCG? They've told you something. It contains live. Now this is a very good clue to that. So cells are replicating because it's a live vaccine. So there are high levels of antigens because it's a live vaccine. So this is going to result in a very strong and effective immune response. This is going to provide artificial active immunity. So boosters are not going to be needed. More so, vaccine does not cause disease because it's a weakened TB bacteria. Please remember, it's not a virus. You write this so very often. Makes me very, very angry when you suddenly switch from virus to bacteria, bacteria to virus. They're two different things. So, and because it is freeze dried, it was easy to transport and store and deliver to the vaccinators, the people who would do the vaccination program. Live vaccine, so cells are replicating, increased levels of antigen. Why? Because you see, I mean, if in the injection you gave in 1 million weakened bacteria, they are replicating, so there'll be 2 million in 20 minutes. So increased levels of antigen, it will result in a strong effective immune response. It will provide artificial active immunity. Vaccine does not cause disease. And freeze-dried, so easy to transport, taking from one city to the other where the vaccinators are going to use it.